Well, okay, everybody. I really appreciate you joining us today. We've got a conversation and a great panel related to cancer R&D, the second wave or the third wave or the fourth wave or whatever wave we're on right now. It's a great opportunity to cover probably the hottest topic right now in research and development in terms of what's going on with cancer drug development, because there's been so much activity here. It is kind of confusing, and I'm sure we're going to get a lot of illumination here from everybody on the panel related to what's coming up. That's probably the most relevant that we're going to be seeing on the near-term basis, but also further out and further down the line, what we're going to be seeing in terms of drug development in cancer and some of the new technologies that are being applied here. I'd like to introduce everybody on the panel. I'm going to start with Andy Plump, who's the Research and Development Chief of Takeda. Andy is our sponsor for today's conversation. We appreciate that. We're joined by Aviv Regev, the new Research and Early Development Chief at Genentech, coming over from the Broad. Probably the newest person here on the panel as it relates to getting into drug development on the corporate side of things. So very interested to hear her opinions. We have Jay Bradner, who already came over from the academic side earlier and is the head of NEBR at Novartis in terms of running the early discovery development type work that they do at Novartis. We're also joined by Rupert Vesey, the head of research and early development at Bristol Myers, and Dean Lee, the new president of R&D at Merck. And several new positions here. I mean, Rupert, you came over from Celgene, and a lot of people taking a kind of a new kind of an approach, or at least a different one from what happened earlier at the company. So let's get this going here, because there is so much going on in terms of research and development in oncology. Trying to make sense out of this could be very confusing, because we have protein degradation going on, drugging RNA, gene editing, checkpoint combos are coming up, new checkpoint inhibitors, TIGIT, LAG-3, TIM-3. You've got the tumor microenvironment. You've got new initiatives on artificial intelligence and machine learning, and a lot of big questions related to how this is all going to pan out. So Aviv, as the newest member of the corporate research and development field, I'm curious how you view this whole thing in terms of where you're jumping into it and where you see the most realistic advances coming fastest.
I think, so first of all, thanks for having me here. And it's fun to be both with old friends like Jay and newer friends like Dean. I think there's one of the exciting things is actually the breadth of opportunities. And I think that also highlights the main challenge that we will have to pose, and I'll try and make a point related to that. So there is opportunities in everything that you said. It's not that one of them is the silver bullet and all the others are not going to pan out. I think what's exciting to me and for many people on this panel is the fact that we can see both the potential of the degraders and of the cell therapies and of the combinations and of the bispecifics and of many other things, RNA therapies and so on. It makes almost everything a legitimate target if we only knew which one to go after. And at the same time, actually, our ability to do human biology is much more enhanced than we had it before. For example, you mentioned the tumor microenvironment, the ability to analyze patient samples now, both from clinical trials and from the general patient population is so much more advanced than it was before, that it gives us a much higher resolution view into the tumor microenvironment. And so to me, all of these things are great opportunities, but they're also a great challenge, because humans in general, when they have too many options, they often tend to flock either to the same things because they all appear appealing to everyone sort of in the same way and for the same reasons. And by this, we might be missing the opportunity for investing so much into oncology. We'd rather have a breadth of opportunities rather than all narrow down into the same places. And the same thing is, and the second thing is, and I think others on this panel would agree with me, it's still pretty hard to predict what would actually be successful, which combination to go after, which target to go after, what type of state we want for the cell therapy. And one of the motivations personally for me to move, and I think it was also something that we see across the field, is that we hope that that our ability to both generate data at unprecedented scale about humans, about patients, and the ability to go after any target that we would want, because we have so many different modalities at our disposal, has to meet the ability to predict better what would actually be the outcome of the interventions. Where are the greatest opportunities, and are there targets that look like no other targets before? And so for each of us to take a more ambitious approach. And I think that is where computation actually plays a really critical role. So if you look at something like degraders, you know, there's a great understanding of the biology of some examples, but I don't think Jay would dispute that our ability to predict which small molecule would behave as a degrader and which would behave as a molecular glue and so on is still not where we want it to be. We want it to be by design rather than, you know, by happenstance. And there's also examples like, if you think about synthetic lethality, or the ability of combining immunotherapies in immuno-oncology or other microenvironment targeting drugs, there's too many combinations to try all of them. There aren't actually enough humans in the world to try all possible combinations. And so is there an ability for us to predict based on partial information, the information that's missing? And that's really what has happened in other fields in the last decade. And now we're poised to fully take advantage of this. I think everyone senses that. And then the challenge is, of course, that there is a difference between hype and reality. There's a big gap between feeling that if we just put it all together and there was some algorithm, it would just show us the way, and actually having the right data and the right ability to span space that's bigger than all experiments we would ever be able to conduct, and still come back with the, you know, the right predictions, the right generation of the right small molecules or antibodies or other reagents, and the right interpretation of what might actually be going on in the body. But if we actually seize that in the right way, in the right partnership, we could be talking something completely different even in 5 years from now.
So Jay, obviously protein degradation is an area that you spend an awful lot of time in. What are your thoughts about, you know, what Aviv just said related to you know, what we know and what we don't know, and how that influences the field?
Thanks, John. And I do agree, Aviv, that protein degradation still remains a bit of a dark art. But investments that the fields have made in the last 5 years, which are quite substantial, hopefully have led to real learning that can advance this as a very exciting new paradigm of drug discovery. We're not a protein degradation company, but we do have a very strong program within our chemical biology and therapeutics group. And so maybe considering your question, John, even a little bit more broadly, I think we're seeing a renaissance in targeted therapy that is in part directly attributable to a more open concept as to what a therapeutic entity might look like, in particular in the domain of modularity. The advance of targeted protein degradation is to think beyond the sort of monosynaptic framework of a small molecule therapeutic as binds and activates or binds and inhibits. And we now see through the modularity of recruitment, either as heterobifunctional degraders or molecular glues, that a target protein of interest, irrespective of engaging its active site, can be disabled through chemical cross-linking to the ubiquitin proteasome pathway, really opens up your mind, not just to what types of longstanding targets can now be approached with discovery chemistry and therapeutic intent, but what else can we glue? What other pathways or circuits of cellular biology in cancer that aren't amenable to traditional therapeutics might be short-circuited in that way? We are in the middle of a true renaissance in targeted therapy, and I think the chemical biology thinking and those approaches have a lot to do with it, but it's true even beyond discovery chemistry as well. I think the work in CAR T-cell therapy, it's a new modality, modular biologics, the final delivery from antibody drug conjugates, radioligand therapy. This theme of modularity in small molecule and biotherapeutic design, I find very exciting and gives us a great chance to retread some of the highest hanging fruit, but the best validated targets in cancer science, and also to open whole new doors with phenotypic approaches to circuits we have yet to discover.
So I'll just jump in, Jay. You mentioned one of the words that's my favorite, which is circuits. I think the challenge for us in the biology side of the discovery, finding our targets or the circuits or the cell states that we want to go after, is that we have like these sketchy drawings. This might connect to this and this might connect to that. but our ability to really know what would happen when we manipulate something is quite limited. We have to run a biological experiment to see what would happen, and eventually we have to run that biological experiment in a human as part of a clinical trial. And so one of the hopes I have is that the richness of what we understand today is substantial enough that we can do the next generation of experiments such that they would really allow us to become more predictive. So we would know which circuit we want to go after. And then the ask of the chemist becomes not just difficult from, it remains difficult from a chemistry perspective, and that requires its own innovation, but at least we know in a deeper way that that's a good place to look. And that might also take us beyond our intuitions. Today, we're driven a lot by a combination of our intuitions and a small search space. This is as many experiments as we can do. We're going to dig really deeply into them. What if we could do deep and broad at the same time? There might be a lot of other circuits that we could unlock in this way that otherwise would be missing completely.
Yeah, Viv,
since you brought up protein degradation, maybe I could jump in and say a couple of words, since of course the heritage of Celgene is cereblon modulation, and you know, two of our most important drugs, Revlimid and pomalidomide, are protein degraders through cereblon mechanism of action, and we have made a lot of progress in this field. I see there are some questions popping up in the chat around this area. In our pipeline, we already have 5 cereblon modulating drugs with diverse targeted substrates that are all in development currently for hematologic malignancies. We also have an androgen receptor targeted degrader, which is going very well in phase 1 with oral bioavailability and emerging signals, certainly of pharmacologic activity. So both those modalities are very robust within our pipeline. We also know that in the molecular glue field, there are literally hundreds of substrates that are addressable through the cereblon mechanism of action. We see a number of companies now being formed to track on that work. And to Aviv's point, this is a substantial biology and computational challenge, because many of the substrates that are addressable, some of them are obvious things that would make good next generation therapies against established targets. Others are against targets that people have long sought to drug but been unable to do so. But the vast majority are actually literature deserts. There are many targets for which there is not one single publication in the literature today. And then this becomes a question of mining substantial databases, either through partnerships with synthetic lethal companies or your own internal efforts, as well as the myriad data sets that have been assembled to see if you can pull out the most exciting of these previously unrecognized targets and immediately batch them up to a compound in your library. And I think that's a really important challenge in this field going forward.
Well, when I listen to this conversation, I might frame it in the fact that what we're all agreeing on is there's really 2 axes to potentially think about, which is how, as a broader biology medicine group, we expand what we can do, right? This is the concept of modalities, whether it be bioconjugates, ADCs, whether it be protein degradation. That's all about the tool set. And advancing those tool sets to clearly validated biology is really important. But I also think that what we're also talking about is exploiting what we know and expanding what we know so that we have a better sense of what biologies to actually target. So I sort of separate those 2 issues. What I would say is, if you look in the broader context, you know, you see the words data and technology throughout, and I kind of think about it as data, information, knowledge. Where should we place our modality technologies on? And so there's going to be advancement of both, and I think there will be advances in the academic world, in the biotech world, where they're going to focus on a specific modality or a specific place where data is really important, but I think we as leaders of pharmaceutical companies, what our responsibility is to integrate, because we don't have an exit, right? There's no IPO exit. Our exit is in the patients. And so this concept of how do you balance the expansion of what we can do, because you can get a menu of modalities. You can have one target and now put 7 modalities against it. So you have to have some discipline. But again, the concept of, exploiting what we know. And I would just, posit to all of you that especially in the cancer field, you could imagine that the single largest sort of physiologic biomarker defined group isn't whether you have breast cancer, isn't whether you have lung cancer, isn't whether you have an EGF receptor sort of mutation. It's whether you respond or not respond to immunotherapy, and whether that tumor is sensitive or insensitive. And what's actually interesting to me, and I would love others to speak, because I'm just a cardiologist, but I would say that there's a tremendous opportunity lurking out there, because we have a very strong impact on, for example, the PD-L1, PD-1 field. And what I would say is there's some degree of humility that I have in the fact that I know how our antibody works. But if you ask me truly at a granular level why it works, when we say that it reveals pre-existing immunity, that means I should be able to have the algorithm of what that pre-existing immunity is. I'm not so sure that I can tell what those formulas are. And there's a huge amount of human biology data. So I would separate the issues of what we can do from what we know, and really this issue that Aviv laid out, which is how do we use our unique position, because we are the companies who play at the inflection point of the patient. We hold that data, and our ability to use that data to actually turn it into information, to glean some knowledge and hopefully get some wisdom from it, is going to be a big challenge for us that is unique to us.
Well, I want to get Andy involved in this conversation here in just a second. I do want to tell the audience that there is an opportunity. You have a facility here on your screen to ask a question. If you have a question for the panel, go ahead and ask it there. We'll be screening the different questions as we go through this conversation. If you want to direct it towards a particular person on the panel, let us know what the name is, and we will do that as we go through those. We'll screen that. So Andy, what's your take on this conversation?
Well, I'm having a lot of fun listening. And I have to say that reflecting back on the introduction, it struck me that I'm the longest tenured R&D head on this panel, which makes me quite vulnerable because our lifespans are not typically that long. The other reflection is I have a comment like Dean's, which is I'm not an oncologist. I'm not a cardiologist, Dean. But when you said you're just a cardiologist, that's amongst a panel of oncology scientists. But when one of us gets chest pain, you're more than just a cardiologist.
Well, thank you.
Yeah, but I love having the privilege of working in oncology, having not spent my career doing this, because it's the area where the science is just the most exciting. But what's amazing is when you look at how much we've unraveled around the molecular mechanisms that drive cancer, and how much investment we have, and yet our failure rates and how far we are from really solving this riddle, it's just unbelievable. And thinking back to the comments that Aviv made earlier, I was thinking of an analogy, which is you go to a soccer field, you have multiple soccer fields, and you have 5- and 6-year-olds playing. What do? Everybody, all 11 on the field, are just running after the ball, right? And I feel like that's what cancer is. It's like you have 5 or 6 different games going on of 5 or 6-year-olds, and they're all running after the ball. They're in a different part of the field, but they're all running after the ball. And I feel like we're doing a lot of that. And part of that is because of this massive investment, and we just don't understand where else to dive. And I also really like, Dean, your comment that there's technology and there's biology. And some groups focus on technology. You have some companies that are just ADC companies, and you have other companies that really focus on biology. Our decision was to focus on biology, and what we're going after is predominantly innate immune biology. And not that we understand it better than anybody else, but we just decided to focus and make that an area of intensive efforts for us. And I like unleashing the immune system because you don't need to fully understand what you're doing. You go after an oncogene and you kind of know what you're doing, but you don't understand how the tumor is going to react to you and how it's going to defend itself. Whereas when you unleash the immune system, of course there are unknowns, but you get a whole, you attack the tumor in ways that you don't even understand, which I like.
I'll comment on two things that came up, and one of them will touch on a question also from the chat. I think from Dean's point, highlighting the pieces that we don't know, yeah, we're kind of only as good as our data, or as our ability actually to generalize from this data. One of the comments from the chat pointed out from Atamba Austin that much of our genomics data in R&D comes from white European genomes. And one of the points that Dean said that resonated with me is we have immunotherapies, we give them to patients. We still don't understand when they work. We don't fully understand as an immunologist, oncologist, tissue biologist, you name it, exactly why they work. We understand many, many parts of it though, and we shouldn't, discount the scientific effort that went into that understanding. But we definitely are still not where we would want to be to predict which patients would respond, which patients would have adverse effects, which patients would not respond. And some of it could be in their genetics, not in their cancer genetics, in their germline genetics, in the differences that we have. Also, some people have stronger autoimmune responses than others, and we know that those things might be related to each other. Those types of understandings rely also on the quality of data that we have. And to the second point that both Dean and Andy made, it is extremely important to have the full stack. For me, this was a huge appeal of coming to a place like Genentech. Yes, you can become extremely good at one component of the pipeline, a particular modality, a particular data type, a particular prediction task, an execution of one aspect, one particular part of biology. But this is one of these mysteries where you need all of the layers lined up, everything from the biology to the patient in the clinical trial to the real world data that comes after that, and everything in between to really chain it all together and get to a better place than where we are today. And I think that's our responsibility in the organizations that we run, because there are few organizations that actually have the full stack, and also, Andy, that have the timeline to really see the whole thing through. I would say you should be proud to have the longest tenure amongst us.
Thank you.
You raise an interesting point of even, having now gone over 20 years or more in industry and lived through a whole series of revolutions, if you like, in disease biology. I mean, going back to things like the SNP consortium, the HapMap, gene expression profiling, next-gen sequencing, epigenetics. I see there's a question in here. And when you've seen this cycle quite a lot of times, each time everybody jumps up and down with excitement and says, okay, now we've got, everything's going to become clear now and we'll understand human biology. Of course, in the earlier days, people attempted to buy complete ownership and access of these technologies, or in some cases to all of human genetics and various other things. I think what we learn each time around is that it's an onion, and it is staggering how complex biology is and all the different layers of regulation that exist. Now, each time I think we learn things that are really important, and they have almost always led to some form of breakthrough and next step. And for each of these things, you can point to therapeutic advances that have been made, whether it's in oncology or elsewhere. But it's never quite as all revealing as people expect it to be. And at somehow, and I think we are making progress, but somehow, as you say, we have to be able to put all of this information together and understand it and make sense of it to get to the bottom of what's driving, cancers and other human diseases. I still think we're a pretty long way off from that, except for certain sort of segments. And often you need a serendipitous therapeutic intervention, because once you have that, it actually unravels a lot of the biology to you. And that, I think that's what's happened with immuno-oncology, right? I mean, as Dean said, you've been able now to segment people into those who respond and those who don't. And then everybody's trying to find ways to augment that response. There are a few chinks of light that are opening up with secondary checkpoint inhibitors, and then people are quite focused on different aspects of the tumor microenvironment. I think we'll find other things in there, but it's just going to take, you know, some effort, and sometimes the failures are important learnings as well.
Well,
I'd say
I would like
biology and medicine, I'd say biology and medicine should infuse a lot of humility into people. The sheer numbers are just so high if you do the calculations.
I agree with that.
You first have to take this step back. And the extent of nonlinearity, the fact that you can predict the whole as a sum of its parts. Once you realize that, though, you shouldn't like freak out and walk backwards and say, well, I'll just stick kind of to my corner. But it requires different strategies than in many other places where you might think the next silver bullet or the you know, the brute force will just get me there. Not in medicine, not in biology.
Well, I am curious about one aspect of cancer research and development that's always kind of fascinated me, which is the rapid proliferation of programs around anything that's successful. CD19 works, everybody goes into CD19. BCMA works, everybody goes into BCMA. Not just goes into it, but they have the initial technology and then the subsequent technology comes in. Can you do with a CAR T, or can you do with an allogeneic CAR T what you did with a CAR T? Do you go into this sort of thing? And then you continue to raise the bar, which is great, but it does tend to replicate a lot of research in your field. You see more duplication in your field than anywhere else in the research and development pipeline for the industry. Does that concern you when you see so much going on? I mean, and Andy, you touched on this earlier, just in terms of the amount of activity here, and how do you work around that? Do you simply go in another direction?
Well, I mean, it's good and bad. I think, you know, part of it is a reflection of just it's practical. There's a lot of money in the field. There are very few targets to go after that'll work. And in most cases in oncology, you have gaps, response gaps, durability gaps, cost of goods gaps. And so you can come in with an incrementally better therapy. I think it's bad, just to be very honest, because what it should do is it should drive competition and pricing competition, and it doesn't, which is, I think, the gap that we have to figure out how we solve, which is how to take innovation, reward innovation, and then you know, find ways to reduce overall healthcare burden costs, and then take that money and then pump it back into new innovation, not just the same old innovation.
John, I'd just add to what Andy said, because I see it the same way. It really is a matter of balance. In some respect, having more than one CAR T cell therapy for CD19, having more than one PD-1 enables access to medicines at each stage of the game. There are real and perceived barriers to doing combination work with pipeline medicines at other companies. And sometimes having access to such a medicine expedites the innovation process, allows us to deliver more quickly. In the fullness of time, as Andy provocatively speculates, redundancy of commercial products could lead to better access and better pricing. That has been elusive. But this must be in balance. And the problem that we see right now, especially in a week like this with JPMorgan, is the radical proliferation of fast followers without meaningful differentiation in the drug discovery space. There is just an unprecedented influx of investment into biotech right now. And so much of that is disproportionately going to the 20th undifferentiated CAR T cell therapy, if not the 200th. You know, 14 months after we reported the chemical solution to inhibition of SHP2 through intramolecular allosteric glues, the first patents started to appear from the fast followers. And, you know, I understand that. And a little bit of that can improve drug access. But it's fair to say that the sector is way off balance right now, driven, I believe, by, as Andy has already shared, a lack of innovation in target discovery, a lack of delivery of highly validated targets, emergent discrete therapeutic vectors from the cloud of circuitry, as reported. There's a small number of targets and a massive amount of investment. But secondarily, and this is where I think all of us are complicit with this, there is a market for this fast follower research from peer pharmaceutical companies that has contributed to this storm. My best hope is that there are innovators out there with unprecedented access to capital who really want to move gravity and who have the discipline to curate their portfolio, as we have painstakingly done at NIBR, to 300 projects that reflect just the highest hanging fruit or solve real challenges in access to medicines in development stage.
Well, I got to ask you, Jay. I mean, Novartis just spent $650 million to get a hold of its own PD-1. I think it's going to be the seventh one. It's certainly perfectly good PD-1, but it does tend to replicate everything else that's out there. I mean, you've got PD-1, and Lord knows, I think everybody on the panel would say that in terms of where it works, you know where it works and so on. But isn't that part of the same problem when you have the same sort of investment on the part of Novartis, where there is such well-defined biology that you're kind of getting into the same thing, but also highlighting the fact that there's still not going to be any sort of price differentiation when you get through it?
Thanks, John. We actually have two PD-1s, as I think you know and wrote about, neither of which was invented internally. As PD-1 has such potential across so many histologies, it's a unicorn. It's a once in 3 generations target that we still have a total lack of understanding. It's human phenotypic screening at its finest. But it is a true platform of a medicine. It is a paradigmatic medicine, a medicine upon which so many other medicines must be built. And you know, Dean's a good friend and an amazing and generous scientist, but there aren't enough hypotheses in the world that can be tested with the availability of 1 or 2 or 3 companies' PD-1s. There are strong commercial rationales for a company like Novartis, so serious in solid tumor oncology, to have access to a PD-1. But at least in R&D, we see this as a must-have medicine to build future, standard of care around.
Jay, I mean, your point about the excessive following and so on, is completely legitimate, and I wouldn't dispute it at all. But your second point about PD-1, which obviously is a broadly used medicine, that does go into considerations when companies pick up a program that maybe they were somewhat behind on, but they have a very strong therapeutic interest in that area. And that's particularly true in oncology, where combination therapies and the cycling of people through therapies as they fail on one modality against a target and then respond to that target again when delivered in a different way. It's not quite as mindless as you might imagine. People do think about this carefully. I think certainly in companies like all of ours, if we're miles behind and the thing's totally undifferentiated and there's no opportunity for unique, valuable combinations within your own portfolio, you're not really going to do that, because it's enormously expensive and it just doesn't make any sense. That may not be true in small companies outside that are spooling up and trying to play catch up and attract investor money, but I think we should at least stand up for the fact that there are some rational thinking people in the companies that we work for.
I mean, I agree. I mean, it's good and bad. And I think if we step back and, all of us come into this from a science and a medical background, and we ask, is it best for patients? Right? I think in the end, it probably is. I mean, like you say, Jay, PD-1s I don't know how many indications are approved. Dean, you probably know this, but it's probably approaching more than 3 dozen, right? And there are probably more out there for monotherapy. And the opportunities for combination therapy are probably, hopefully, geometrically more than that. And so in the end, it's probably good for patients.
I want to just circle back to something I think Rupert said, which is how the advancement of science is, none of us would call it linear. In a large scale, there's a slope, but it's interspersed by big leaps. And I think Jay sat there and said, well, PD-1 is one of those big leaps. And so I just want to come back to some points that I think is important, because I do really think that the lens that we can bring to the field, right? We have to ask ourselves, why is wealth aggregated in pharmaceutical companies, and what is it that we're going to do with it? And I just would play off of a little bit of what Rupert said, which is, you know, I think of any successful or unsuccessful drug candidate, those actually teach us more about human biology than oftentimes the body of knowledge that led to the initial concept of the drug. So the interest in immuno-oncology gets back to what I said from a, not from a market standpoint, not from a commercialization standpoint, but understanding what that interplay is, not how it works and why it works, because any therapy that is out there has to now paint itself on the canvas of that. And that canvas is very important for us to study. And I just want to bring back that concept that we are the companies who do human biology at scale. And our ability to tap that along the lines that Aviv spoke about is critical. That is what differentiates what we can do. And the second sort of thing is, I got asked this question about AI machine learning at one of those, symposiums that Merck, those investors meeting. And I said, anyone who tells you that AI machine learning is something that you put on your laptop that suddenly gives you an awe-inspiring insight into the world is sorely mistaken. The concept is, can you take large data sets as curated and collated as possible? Can you take that information? Can you find appropriate associations? But then at the same time, can you integrate it with experiment biology and experimental medicine? And we are the companies who will be able to integrate those approaches that Aviv spoke about. And I just want to emphasize, that's what we as industry leaders can do that I think very few other people could do.
So I just want to use this, Aviv, I'm going to get right to you, but I'm going to flip it right to you, but I just want to use this as a kind of a, dividing mark, because there are a lot of questions in the chat file related to AI and machine learning. And I know, Aviv, this is your area of specialty, but I would like everybody to address this in terms of, because there's all this prospect of advances and everything else, and questions about the data and so on. But there's a tremendous amount of interest, Aviv, in where this is headed and what you can do now versus what you're going to have to wait on later on. So tell us your own perspective on that.
Okay, so I'll try and cram a lot in a quick answer. The first is that I'll distinguish three different things that computational approaches, and I say that more broadly, machine learning being particularly powerful for us now, but applied math has advanced a fair bit in the last decade as well. The first style is what we call prediction. Say you're thinking about the, antibody space. Here's a target, and here's an antibody, would the antibody bind the target? And of course, there's richer biological questions to ask than that. The second is what we call generative models, which in my toy example, if this is the target, please generate a sequence of an antibody for it that would bind it with particular desired properties. And the third is what we call interpretation. And that's really the gold that Dean was referring to, which is the antibody shall bind this target because, and here are all the, the structural elements, the hydrogen bonds, the protein-protein interactions, the works. That's a really simple example. As we go to things like the circuits that Jay referred to, we're in worse territory, but all of our work is one of these three categories. Either we're trying to predict something, or we're trying to generate something, or we'd rather do both of these things and interpret them. And as you move down this road, it gets harder and harder. The second point I'll make is that all of this is always an iterative cycle. Data is not static. It's not like, here's the data, we shall first analyze it, and then we will have some prediction, we'll do some experiment, and we'll move on. But rather, there is data, and there is the ability to experiment. That is what is so special in our field, in our area of both biology and biomedicine, as well as chemistry and many of the associated fields to it. We have the ability to intervene in the system. It's either an experiment in the lab, or it is a clinical trial. But we introduce these interventions, we collect more data, and then we analyze it again, and we make new predictions, generations, interpretations, and we cycle again. making that cycle better, faster, bigger for the same amount of resources that we have is that inflection point that we're at. So that if before we could either do a lot of digging in a narrow area, and that's why maybe we flocked so much in the soccer game around the same set of balls, now we can take and have sort of the same level of information, but across many things simultaneously, which also allows us to understand how they relate to each other. But we will never be able to exhaust all these experiments and data collections that are imaginable. And the data will never just, to Dean's point, be given to an algorithm and given back with a complete answer and we have the answer to all mysteries. It's going to be this iterative process of back and forth and back and forth. And the trick is to make it tight and strongly connected together. And there was a question specifically in the chat about random experiments. I don't want to belabor this, but the general idea is that when the space of possibilities is particularly huge, human genetics, interactions, combinations in synthetic lethality, and so on, sometimes, and you can do a certain scale of experimental exploration of particularly large kind, sometimes the best way to place your bets is at random and not be smart. You can be smart in the second iteration after you've collected something, but not necessarily in the first one.
Hey, John, as the question asks about sort of concrete shovel-ready applications of AI, let me offer a couple of thoughts. It's actually a pretty small list. You know, when even image-based AI works in a car, I mean, I drive a Ford, so we don't have that technology in my car. When you take your hands off the wheel, you know driverless cars are working. And it's fair to say, not living in that moment now. So even in the much less advanced space of drug hunting, AI is pretty new, but it's also pretty exciting. As we've prioritized our work in AI in drug discovery and drug development, a lot actually rests upon operational efficiencies and operational execution, predictive analytics, where AI works really, really well and can lead to, we think, meaningful even margin expansion. In drug hunting, generative chemistry is still very new. We have excellent, we believe, two-dimensional molecular representation, but three-dimensional molecular representation is quite elusive. There we will make the investment. Predictive pharmacology, shovel-ready, works pretty well. The trick is to get the old chemists like me to actually use it. Search. I don't know what the search fields are like at your companies, but we have an awful lot of data that's unsearchable, and we would love it to be so. Natural language processing to try to bring the knowledge base of a large community like our, 6,000 drug hunters into a single hive or collective. And then finally, in target discovery, we see some applications like cell painting or phenotypic screening, multidimensional nucleic acid detection, which Aviv's a true world leader in. But it's fair to say, even with knowledge graphs, these are suggestive methodologies that are really just figure 1A of a long story of target discovery and validation. So the AI drug is a little bit elusive today. And what I would love to see from the external world, which is, I think, more likely to bring real innovation even than our great companies together, is to really apply some of the highest hanging fruit, the challenges around three-dimensional molecular descriptors, not to the fastest kinase inhibitor ever discovered, but to really get us into brand new territory. KRAS G12V, that would excite me.
Yeah, I think, Jay, I agree with you that there are sort of these different places where things can be applied. There's a couple of other good examples. I think in biomarker work, particularly digital pathology and imaging, there are really remarkable opportunities and ways of finding responders and non-responders. And I know there are health systems where all pathology is being digitized, like in the UK and so on for this purpose. The other area that's been very fruitful for us is a sort of, and Jay may say the same thing, but it's a sort of unique ecosystem, which is cell therapy, because you have you have an amazing ability to make the drug from the patient, to generate translational data from that patient, to generate clinical response data, and then you have vast amounts of manufacturing parameters. And by integrating all of those things into a system, you end up with a huge number of data points, and you can find through machine learning algorithms all kinds of important things that you would never have previously predicted that are important to the performance of cells and the selection of patients and how you manufacture them and so on and so forth. It's just a really nice system where you have all the data in one place, and it's incredibly tightly associated. I think, you know, what would be great, maybe Aviv's going to do this because she's going to have a much longer career than some of the rest of us on here, but you know, is actually to go from soup to nuts in terms of AI. I mean, we just, you know, put in place a relationship with In Citro. I'm sure Aviv knows Daphne Kohler really well, so we're excited about that because that's sort of what she's trying to do, you know, is make everything work through large data and AI type of strategy. And you know, maybe doing that in a fresh field environment like that will be successful. Jay talked about the challenges in big companies, particularly when they've been through multiple integrations and mergers, of getting all those data silos together in order to use all the information. And every time we do one of these events, you know, there's 3 years worth of sort of harmonizing IT systems at the most basic level before you even get to integrating all of the data. I mean, I can think back over the companies that I've worked for, like GSK, that did, you know, all of the target class, you know, chemobiology. I don't know what's happened to all that information. At Merck, we had, you know, reams and reams and reams of gene expression profiling data from the time that we had Rosetta, that Andy will remember well. Maybe Dean has that on his laptop, I just don't know, but you know, I'm not sure where all this is. And so I think over the years, you know, we lose a lot of the information in larger companies, and we never quite catch up with it. So, but you know, maybe in an environment like Genentech, you can pull all those things together and do, you know, AI discovery from beginning to end, and I think that would be wonderful if that can be done. We're certainly trying to find ways to do that.
Let me jump in. I wanted to make a comment back because you're referencing something that Dean said, Rupert, which I liked, which is how important learning is in our failure. And there's an aphorism in sports. You know, the great sports managers say there's two outcomes: there's winning and there's learning. And
yeah.
But we're terrible at that, to be honest.
Right.
Even within companies and across companies, we don't share data, and so you don't have that opportunity to learn.
I agree, Andy.
And then I'll just emphasize the point that you, Aviv, Jay, are making, which is that there are selected examples where these advanced analytics like AI and machine learning can add value. But the reality is that, you know, in between that and pure intuition, there are other ways to leverage data that we don't even do today. Simplified ways of using data, you know, to maximize and optimize clinical, you know, the most inefficient thing we do, let's be honest, is how we run clinical trials and how we recruit patients. And there's so much opportunity there that doesn't require these advanced analytics. I'll just tangentially, my son graduated from MIT with quantum computing and machine learning degree, and now he programs robots. I won't go into, you know, the kind of confidential pieces here. I said, oh, it's really cool. So the robot's a learning machine and it's using machine learning. He goes, no, all the algorithms are just, you're basically not using. So these tools are just not, I mean, there will be applications and they're certainly going to come, but I think even short of applying artificial intelligence, there's so much that we could be doing around data that we're not doing today.
If you can just indulge me for 1 second, I'll be really quick, but you know, in all the years I've been doing this, one accomplishment that I'm actually really proud of, not the only one, but one, and this may make you laugh, but is actually to get our organization to accept objectives where we get rewarded for making no-go decisions. I mean, genuinely rewarded if they are science-based and informative. I had previously never worked anywhere where anybody would accept, particularly, you know, at the compensation committee level, that they would reward the company for making a negative decision. And of course, that just reinforces what you just said. Nobody ever learns from a negative decision, and everybody tries to keep things moving because they want to say that it was positive. And you know, I think if we could be more stringent about these kinds of things, we would learn a whole lot more. And then your point about sharing, Andy, is a good one too.
To Andy's point on machine learning, I was actually careful when John first asked me. I said it's computation as a whole. Machine learning is definitely an important component, and it has shown us how much can be generalized from data and how much sometimes you don't actually need to know the underlying details in order to still get what you want out of it. But at the same time, the radical change in data platforms and in how computing is done changes what we can do in ways that can be impactful irrespective of the algorithm. So all of these things play in. I think it's really the whole, if you think about it linearly, which it's not, but if you think about it as a line, starting, with initial biological discoveries and ending with a drug in a patient, every single step is up to being transformed. And some of them, and it's almost backwards. It's almost like the things that are latest in the process. People mentioned the biomarkers and some of the predictive analytics and so on. The design of clinical trials are sometimes the earlier to be impacted. But the really big promise is if we impact the things that we discover that we want to target, be it classical targets, molecular ones, or cells that we're trying to get to the right distribution in cell therapy and have them behave in unexpected ways. These are the things that would change dramatically what we have, not just how efficient we are, but qualitatively what it is that we have.
I really want to bring Dean in on this conversation as well, because obviously your predecessor, Roger Perlmutter, the first thing that he did was join Daphne Coller's board of directors. And one of the things that she emphasizes is, how much she had to learn about drug R&D, which is why she turns to people in drug R&D. And there is kind of a disconnect in a lot of different respects when you see some of the AI organizations and some of the things that are being tried on R&D. So let's get your perspective on that.
So I would make 2 or 3 points. The first point I would make is I think we as a biopharmaceutical company have to understand how we're uniquely different, but also the threat that's coming to us. When we talk about our data, I don't know if you guys remember Raiders of the Lost Ark, the very end where they're packing the Ark and it goes into the warehouse. That's pharma and how it deals with its data. It's really powerful. It's whatever, we've got it protected, Dr. Jones, and it sits there. So that's one imagery I want to give to people. I think the second sort of issue is that when you talk about data, the point of having data is to change what you do. And I just want to emphasize what Aviv said. If you don't have, whenever someone talks to me about AI machine learning in biology, I want to see their labs. I don't want to see their computers. I want to see their labs. I want to understand how they took that data and immediately changed how they did experiments and did those experiments. I need to be a little bit careful and not respond to the in vitro comment directly, because I think some of you know that I'm a founder of Recursion, which also plays in that space. And I like to say that as much as I appreciate the sponsorship and mentorship of Roger Perlmutter, who's been fantastic. We have choreographed this. Him moving, we make a joke, game's on. The game is on. But the issue that I would just say is these other companies in different, whether it's molecular, whether it's phenotypic, and I would just reemphasize, Jay, phenotypic, empiric drove medicine. We start to believe that we learned about molecular pathways and then started doing Nodo. And you made an interesting comment. In some sense, Pembro is from a phenotypic screen. And that's the unusual biology that you learn something from. And essentially what AI and machine learning is doing is weaponizing that. And the fundamental issue for us, it's in molecular design, it's in omics, it's in clinical trials, it's in all of those. And the fundamental question that the pharma has to understand is, are you going to do that? Because if you don't, your 125 years of advantage really is the last 5 to 10 years. That's where your data really is. The ones that are 30 years, we can't quite find. And these younger companies that are coming, every piece of data is being curated and collated and used. And someone might come in from the information technology side and begin to wrap up those different sort of basic infrastructure. And so for us, we have that integrated full biology view, but if we don't pay attention of what's happening in this field, our advantage of having 100 years, 130 years of experience of doing human biology could look a lot different 10 years from now.
Okay, so I know this conversation could go on for the rest of the day, which is great. I think we got a lot of ground covered, but we're going to have to wrap it up. We have run out of time. I really appreciate everyone joining this panel. I really appreciate everyone in the audience taking time out. It's been a fascinating conversation. I think it underscores just how much interest, just how much money, just how much intelligence is going into cancer R&D. And obviously there's going to be a ton more coming over the next few years. So this is going to be a topic that we'll get back to in the future. I'd like to thank everybody individually on the panel. Thank you very much for joining us. It's been a great conversation. You've been a really interactive group. I love it. And again, everybody in the audience, thanks for joining us. We'll be back next year, I'm sure, with the same group and the same kind of conversation about the other things that we didn't cover. But thanks very much. And with that, I'm going to wrap it up. Thanks to Takeda for sponsoring the conversation. Take care.
Thank you.
Thank you.